the pre-lecture lecture. Well, plant responses to internal and external signals. So let's just go ahead. I'm going to play this as a YouTube video you can find almost anywhere here. A lot of similar ones. So you have that imbibing, right? There's an imbibing of water into the hypocotyl. See how the radicle comes out. Could y'all tell what comes first, the root or the shoot? You have to look past it. I think if this were your crop, you wouldn't want your corn to germinate with this much difference, right? It has to be much more uniform. Just make some observations. Now tell it's slanting away from the shadow. Y'all ever done this experiment? Put a corn cob in the water. That's all this is. Didn't I hit this issue last time? Or it went smaller? I guess y'all can still see it. So we got, you know, what came first, the shoot or the root? Did y'all see anything in the shoot coleoptile, the front or top edge of that shoot? Okay. So So to get through kind of what I think or believe you would know before coming in here, is that plant hormones, a big bottom line, when we're talking about growth, phototropism, gravitropism, there's going to be a lot of hormones involved, okay? So hormones are simply chemical signals, and they coordinate different parts of the organism at different times. That's why there's a life cycle, right, of senescence. Even humans have a life cycle. The hormones you secrete at 2 are different from 12 and 22 and then 62 or whatever age it is. You know, hormone levels are in flux, and these are genetically programmed for each tree or crop or plant species. So any response, now they tend to be the same hormones, but their ability to respond to the environment clearly is different, because we don't grow most cacti and succulents that would grow naturally maybe in a desert here. And deciduous trees don't grow well in Arizona, right? But nonetheless, it's the same hormones. So there are species differences in their ability to sense these things. So tropisms are often caused by hormones. So this is kind of what we're normally taught. And that starts then from the 1800s. This is the observations. Charles Darwin and his son Francis conducted experiments. These are some of the earlier ones, which is not to say they're the first. I think Carl Linnaeus first came up with that kind of floral calendar based on opening of leaves and flowers. But they observed that grass seedlings would bend towards light only at the tip. Only if the tip of the coleoptile was present. So they hypothesized that a signal was transmitted from the tip to the elongating region. So I cut out the up close pictures because I thought it was going to be bigger. I don't know why. Each time I do this, it does. Man, it's the same thing. But I'm going to try opening it again. I don't know that it, oh, there's the line stuff. So what part of the grass coleoptile senses light, and how is the signal transmitted, right? This is the question. If we're seeing that there's a response, something has to detect that light, something else has to respond, okay? And so where is that happening? So the general observation we look at in the control is that light hits this and it bends, great. And what we notice is that the illuminated side of the coleoptile takes the bend and these stretch. So when we look at that, we see where there's growth. So what they did is they cut off the tip on one, and on the other, they covered the body, right? So this will tell us what? Looking at these two, this will tell us if it bends here, it's at the tip. If it bends here, it's on the body. So when they cut the tip, there was no bending, therefore there's no receptors, or at least whatever the stimulus is, whatever the sensory organ is, it's not here, because there's no bending. But nonetheless, when we did cover it, it's still bent, and it tells us the sensory as well at the tip. So just to kind of prove that, we looked at, they covered it up, kind of opaque. So we left everything intact, and we just covered the tip of it. Nothing happens. I say we, but I mean, so. And then they covered it again, but with a transparent cover. And so it's not to touch temperature, but whatever it was signaling, it was sensing the light. So it had to be visible to the light, one, and two, it must have been at the tip of the coleoptile. So that was Darwin and Darwin. Then Boysen and Jensen came along. And well, how does, because what is actually doing the bending? The body, right? But yet there's no, we know there's no signal there. So they get the agar. I don't know if you know what agar is or agar is, but it's like a very thick jello. But, and then they got kind of a plastic, and they put a barrier there. So this is a permeable, like gelatin, a permeable barrier, meaning that whatever is up here can be sent down and whatever is down there can be sent up. And likewise here though, there's they put a barrier, but it's impenetrable. Nothing can get through it. So we're going to kind of mess around with the communication along the cell. And sure enough, of course, you can see now that if the signals can get down, the whole thing bends. So we know from this experiment, it's at the tip of the coleoptile, and we know from this experiment that whatever happens at the tip of the coleoptile gets, it's a signal that's transduced to the bottom. And up here, we have no bending. So whatever happens in the tip is important to the bending of that plant. Very simple, very beautiful experiment. Learn a lot from these simplicity. So Boysen and Jensen demonstrated the signal is mobile and a chemical substance. So that's the experiment I just described. So Fritz went, extracted the chemical messenger for phototropism, and Auxin was found in modifying the earlier. So what he did, right, is he excised the tip and he placed it on an agar tube, again, a gelatinous or a thick gelatin tube. This, and I don't know, again, if you know, when we talk about electrophoresis and things, gelatin is porous, and so things can diffuse across it. And so you can see here, the agar block is being infused by the diffusion of this Auxin chemical. So the control is they cut the tip off, no bending. The control block with no chemical, just to simulate that. So you have two controls here. And then they put this block with the chemicals. The tip is not there, but the chemicals are. And then we have it cut in half. And what do you notice is happening, if you can see right here? Wherever there is that pink, the chemical is diffusing into the plant, and it always bends in the opposite direction of that chemical signal. So here's the pink on this side, it's going left. Pink is on this side, it's going right. So now we know that there's a chemical that's produced in the tip that if it can diffuse down, signals somehow to these other cells to bend. What is that bending? Well, we know now that it's a cell elongation. And as Austin said, Auxin causes shoot elongation. And so here we have the disproportionate elongation of cells growing while other cells are not, and the natural effect is a bending action. So it's not like the leaves you mentioned, how leaves can just drop or fold up. Now that's a whole different type of movement, right? Do it like the pulvinus at the base of the petiole. That has to do with rushing water into and out of cells really quick-like, and some ions. So that's a different kind of movement. This is an overall growth movement, a tropism, affected by Auxin. So I've kept this in there just to show, and you can refer to it later, but the various actions of the various hormones is worth reviewing. Usually you study one or two because one or two interact in a system that you might be interested in. But just briefly, Auxin refers to any chemical that promotes elongation in coleoptiles. So indoleacetic acid is a common auxin in plants. And it refers specifically IAI here. You can have indoleacetic acid. So it's like a vinegar, but a little different, right? Acetic acid is vinegar. You can also have IAA negative. You can have IAAH. All these are forms of auxins. These are important because different receptors recognize and are that have an affinity, that is an attraction to different ones. Check it out. If you're negatively charged, right, then you're going to be attracted to a more positive environment because opposites attract. And if there is a protein receptor in the membrane of that cell that lets you in, you're in. Others, the more neutral, and others still, this is the acid carry the reduction, the reduced form, and it can donate those protons. These two are kind of go back and forth depending on that hydrogen proton. So it kind of relies, there's a little bit of chemistry there, but I want you just to make note that all those are Auxin. And we're not going to spend too much more time here. So what tells the Auxin where to go and how does it perform its function in order to play? So now I got to quickly go back to the beginning of mine. There it is. Okay. So what do we know? We already know what the environmental signals are. We know it's in response to light, and we know the location where those signals are received. Does that make sense? We know that it's, we're looking strictly at phototropism. If it's gravitropism, you can extend the same thing. The stimulus is what? The force of gravity. It senses gravity. Now that in itself is the what they call statoliths or stratoliths, the rocks and orientation of those within cells. We maybe get to touch on that. So what we're interested now is at the cellular level within the coleoptile, let's just say, how does it detect out light and how does it cause growth? So we want to connect those two ideas. The idea that we know auxin is the cellular signal, the hormone, and we know light is the stimulus. How does the light stimulus result in auxin being distributed? And how does that diffusion occur? And the answer in short is something called signal transduction. Signal transduction is a very common thing to get your master's or PhD in. If you can learn and elucidate a signal transduction pathway, hot dang, you're in good business, all right? There's a lot of them. At least me, I won't speak for you, but I was really, when I got into biology and even genetics and how things work, it's just, I was really expecting this paradigm, kind of Mendelian. You know, it was as simple as you learn the DNA has nucleotides, it codes for these codons and mRNA that go to the ribosome, and if it's an AUG, it codes for methionine, and it's a CUC, it codes for leucine or something else, right? And oh, look, there's the protein, and the protein by its structure does whatever. Done, right? Like, that's how life works. That's why it's a central dogma. And since about 1973, we've been learning everything except that. I mean, it's just not that straightforward or simple. It's very hard, actually, to find a textbook anymore that teaches that straight linear. This is very old school in the way I've approached it, to be honest, because we've learned so much more, and it's crazy. So there's no single thing that we can say, oh, all things happen like this. This always happens like this. There's very few of those things, unless they're really fundamental processes like leaf development. That's common to all plants, or root development, which is common to all plants. But the individual traits are exactly that per species. So you really could study signal transduction pathways using bioinformatics and or molecular biology techniques. So I'm going to go through a signal transduction pathway generically, okay? That's what these slides are. So I'll be a mixture of words and pictures. So plants have cellular receptors. So when I say cellular receptors, what does that make you think? That detect their changes, that detect changes in their environment. What What do you think of when you think of a receptor? What's the picture you have in your head? I know you never had one, right? Now what is it?
They're like embedded in the wall or something like that.
That's something like that, absolutely. Not the wall, but the yeah, the fatty stuff. Phospholipid membrane, right? Yeah. I think of antennae, like on the house, you know, or on your television set, antennae stretching out. Okay. If I drew, I'm gonna erase, I didn't want to erase that yet. So if I drew a phospholipid membrane, here's my phospholipid bilayer to my cell, right? And the cell, you know, kind of goes along. So the cell is coated in membrane proteins. And those membrane proteins are sometimes, you know, they're channel proteins, and they have a little hole so things can pass through. Sometimes they're, you know, they have a specific shape for only a, you know, a specific signal molecule. Oftentimes, they have a shape on the inside and some funky receptor shape up here, but there are just all kinds of proteins in the cell membrane performing myriad of communicative functions. Okay, if we can get the structure of important cell membrane proteins, boom, one of the ones we're going to talk about today is very recent, as a matter of fact. So these are membrane proteins. And so when we talk about receptors, I like to think of these as antennae. What do antennae do? They receive signals, right? Radio signals. You tune your, I mean, I know it's like now really going old school. You turn the dial, and once you pick up a certain frequency, you get the signal and you hear the message. So the cell in its DNA, in its nucleus, and here's the DNA, right? This is where the action actually goes down. This is what we're curious about. How does this respond? Well, if you have a receptor for a particular signal, this is where I want my rainbow colors. Blue light, red light, where's my yellow marker, orange marker, black marker, all the colors of the sun, right? So all this light, a lot of it, actually I'm gonna use black for this. A lot of it, well, not in the case of plants, but everything else, hit and may bounce off, okay? But if you have the receptor for red light, receptor for blue light, you got the message, and you send that message somehow to the DNA. That's signal transduction in its simplest. You have a receptor, gets the message, and you transmit the message into the cell here. That's when it is for any kind of protein signal, such as Auxin. So Auxin must bind to a receptor, and then a message is given. Each cell has its own machinery, mostly pretty similar. Now, if that message, and there are in our, I'm not going to go back there, but in the line of hormones, some of those are more steroid-based, which is a fat-like. Now, if it's fat-like, Auxin just said it. What is this membrane made of? Phospholipids, bilayer. So it's a fatty kind of cell. Like dissolves like. So guess what? That message doesn't need to go through a receptor. It might come straight in, right? Because it can come through the membrane because itself is fat. All right. But we're mostly interested right now in Auxin, which does require a receptor. So for a stimulus to elicit a response, certain cells must have the appropriate receptor. Stimulation of the receptor initiates a specific transduction pathway. For instance, as we get to light responses, which is what I want to talk about, potatoes, we've seen, at least in my house, I keep them in a drawer in the dark. And so I see this because I'll forget about them until I smell them. I'm like, oh my gosh, the potatoes. They smell rotten death. You ever let your potatoes go rotten in your house? Good job. You're eating it all up. Yes? Good. You know, the pain when you open that cabinet. So potato left growing in the darkness produces these shoots that look unhealthy, lacks elongated roots, right? These are morphological adaptations for growing in the darkness, collectively called etiolation. So this is etiolation. I saw this on my grapevines when I was growing rooting grapes in the cold room. It was dark all the time and it was cold all the time. And this is what they did. When it was time, they begin to get desperate and etiolate it. After exposure to light, though, a potato then can undergo changes called de-etiolation, in which the shoots and roots grow normally. And so they begin to sprout real leaves and grow real roots. Same potato once it's exposed to light. What caused this change? Obviously, the light. So same question. A potato's response to light is an example of a cell signal processing. The stimuli. The stages are reception, transduction, and then the response. You ready? Reception, transduction, and then the response, which I don't have here yet. So you got to be able to receive the signal, relay the message, and now, whatever it does, you know that the DNA has to result then in a protein. This is protein synthesis. I'm going to draw a funky looking protein here. That's going to, you ready for this? Change something about that cell. That's the whole point of protein synthesis, is to make that cell do something that it was not doing before, right? Reproduce, grow, take in auxin. So this protein, and we briefly hit this, protein trafficking, but if this was done in the ER, all details left out, right? This could get shipped up here to the membrane. So watch this. You have the signal, the reception of the signal. It was transduced to the nucleus, where it had its effect on the DNA, and it responded by making a protein, in this case, a membrane protein, that said, let auxin in. And auxin actually is recognized by that protein, then auxin comes in. The light signaled the DNA to make a protein, or at least to transcribe the DNA for a protein that said, let auxin in, and now we grow. Or keep auxin out, we don't grow, whatever the case may be for the tissue. So that's in a general sense. Make sense? Kind of. So see the picture of it. Makes sense? So you have internal and external signals. Second messengers are calcium. Calcium is actually a second messenger. You might have seen that in crop physiology or plant structure and function. I'm not sure which class covers it now. CAMP, cyclic adenosine monophosphate, which looks a lot like this. You see it as C, a little c, and then a big AMP. Calcium. These are two good, very typical second messengers. What's the first messenger? Whatever the original stimulus was. In our case, light is the first messenger. So there's a picture of it. Light It's a phytochrome. Are you ready for some chemistry? Do you want chemistry? Maybe not. Maybe so. We'll hit a little bit. I did say it early. All right. So there is a little bit just foundational biochemistry that really helps you to seal the deal in the particulars. But I'll go somewhere in between. I just want to say that these are not unique in small situations. This is canon. I don't know, can you say this is canon for cell biology and transduction pathways. Okay, that's why I'm able to write calcium in CAM. It's not like I know all these intricate details. This is within the world of cell biology and molecular biology, very common knowledge, in other words. So light strikes the phytochrome. This is less common knowledge. Phytochromes are light-sensitive proteins. A very interesting molecule we're going to talk a little more about in a few moments. But think about any other molecule you know that is actually influenced by light. Photosynthesis. And where is it that those photoreactive proteins exist? Where are those photoreactions taking place? That's right, in the thylakoid. Well, in the chloroplast, thylakoid within the chloroplast. And that's right, there was the whole ETC, electron transport chain, between the two photosystems. So you had photosystem 2, photosystem 1, light strikes them. I know I'm doing a lot of assuming, but I think it's been probably a while since you've had photosynthesis, but you might recall that there is a light harvesting complex at the heart of that photoreaction center. So what I want you to imagine right here, you ready? Let's draw on our imaginary whiteboard right here. So you have a cell. Within the cell, you have chloroplast. That chloroplast has a membrane. Within it, you have thylakoid. So we're going to blow that thylakoid up. And within the thylakoid membrane, you have membrane proteins, just like that, except that they have receivers for light. And it's not that they receive light, they're simply influenced by light, meaning that when light strikes, whoa, right? It's kind of like you change all of a sudden. I want you to think of that because that's exactly what happens. They get excited. But imagine being excited without anywhere to go. You ever been at a mosh pit? Got to get in there, right? So you're trying to move, you're jumping, and your one movement is going to influence everyone else's movement, right? Or maybe pool balls are a better thing. So you have them nice, tightly packed, right? And you hit just one, and you want that energy to spread all the balls, right? Same sort of deal. You have a photoreceptive protein embedded in a membrane. When light strikes it, it gets excited and jumps, but it doesn't have anywhere to go. What actually jumps? The electron, right? It gets shot. And as soon as that electron's lost, it's replaced by another electron from water splitting. All right, that's all the photosynthesis we're going to get into. But I wanted to draw it because I think we've been exposed to that. So if you could just imagine chlorophyll, which is where I was trying to go, chlorophyll is like an antenna receptor. When light strikes chlorophyll, remember the action spectrum, the light spectrum? So when various light wavelengths strike that chlorophyll, it excites it, and it actually loses an electron, and everything around it gains those electrons and passes them right on down. That's a chain reaction of events, all due to what? Light striking chlorophyll. So this is where it gets kind of cool, I think. So phytochrome is very simple, but where is phytochrome located? It's a pop quiz to see who's not zoned out. Yeah, that's right. It's in the cytoplasm, not in the thylakoid, not in the chloroplast, not in the nucleus, not in the membrane. It's in the cytoplasm. Crazy. but true. And phytochrome, for now, let's just say similar to chlorophyll, gets excited by light. But it doesn't give off electrons. Instead, it uses the energy of light, and it is just changed. So a word you want to learn, I want you to say is conformational change. Say it. It's fun. Try it. Conformational change. It's important because there's only a few times, actually it's not, it's a lot. But I don't want you to have to just accept fact. I want you to understand that if somebody were to poke you right now, if I walk up and poke you right now, you're going to move, right? That's what it is. When light strikes it, something changes about it, and it creates this transduction pathway. All right. Why did I draw ATP? So I want you to assume and see that this is a kinase. K-I-N-A-S-E. This is another very popular. I mean, if I were to give a quiz or a test specifically on this, I would ask you about what are the steps of a signal transduction pathway? 1, 2, and 3. Be able to name them. All right. Reception, transduction, response. Okay. Kinase. What does kinase do? This one's important because I've said it a few times, this term. Kinase phosphorylates and dephosphorylates. So this is kind of background knowledge to understanding, because I could just say this happens, this happens, this happens. You're like, great. So those things happen. What are those things? This is me attempting to explain what those things are that lead to the big picture. So phosphorylation is the addition or subtraction, if it's dephosphorylation, of phosphate groups. Phosphate groups is PO4, and they are a wonderful molecular tag. You would be like, I mean, this is the first time I learned this, I thought, there's just this one word, and it was a long word, it made me feel like, and then I saw it again and again and again and again and again. Phosphorylation and methylation are two ways in which the largest things in cell biology are accomplished. It's the simplest kind of reaction. Because of the negative charge, these guys repel each other, but nonetheless they're covalently bound. And kinase molecules have an active site within them that recognize, bind to it, and they're able to put just enough pressure to separate it. And so that you now have, this was ATP, right? Now this is a DP, plus we have a lone phosphate group over here, inorganic phosphate. Something's picking that up, and kinase is attaching it. So what just happened is this kinase dephosphorylated ATP and phosphorylated whatever other molecule. That's what kinases do. And they're everywhere, they're ubiquitous. Every cell, every function, every meiosis, mitosis. Now there's all kinds of something kinases, okay? So there's a slew of families of kinases. So you can expect that this is a region of the DNA that is always unwrapped and ready for expression. Why? Because everything uses it. Everything is either phosphorylated or dephosphorylated in order to mark it for a reaction or no reaction. Crazy, but true. So this protein looks the same in every cell, and therefore the DNA sequence that codes for it is the same. And we don't need a lot of them, but we do need to know when and how much and what this is going to be. All right, so we know that some receptors can be excited by light, aka chlorophyll. Now we learned a second one, phytochrome. It's going to change position. And in changing position, there's going to be something to do with the phosphorylation we're about to get into now that you know that. Since by phosphorylation, a series of phosphorylations, the message gets activated and somehow into the nucleus. So look at the response here. Transcription factor 1. It's already present, right? Is it reactive? Not like this. But once that light strikes the phytochrome, which phosphorylates, sends the message, and a series of phosphorylation, dephosphorylation, phosphorylation, dephosphorylation, the transfer of that P group all the way down ends at transcription factor 1. And now that it's destabilized and activated, it can bind to the DNA, and you know what happens from there. Transcription, translation, and in our example, de-etylation, that is the greening response proteins. What part of the DNA? So that transcription factor must be a regulator of greening proteins, and that's the protein that was made in this expression. All right, does that make a little bit of sense? So we just went from light to cellular response in a generic way. There are more details to that. I'm gonna take a breath while you read this. So I did have a lot of transcriptional slides here. I hope I took out, and I just summarized it in this one. Wow. That one little component of transcription that we learned so far keeps rearing its head up. What I'm referring to is how transcription occurs. So we had DNA unwinds, RNA polymerase goes through and all that. But when does that happen? There has to be a promoter, right? And for that RNA polymerase to transcribe, you must be able to bind to the promoter. Well, what's necessary for RNA polymerase to bind to a promoter? Transcription factor. So first, the transcription factor comes in and it recruits the RNA polymerase, and then it goes down. And not that I'm saying it's just one, there's many. But that was the picture we formed back then. Just want to bring to our memory. So some transcription factors increase the transcription of specific genes, while negative transcription factors are proteins that decrease it. So think about auxin again. And how is it that it inspires growth in the shoots and prohibits or in the roots? Well, it's going to be what happens inside that nucleus. It could be blocking RNA. So the receptor for auxin blocks RNA polymerase in roots, but the receptor for auxin in shoots actually binds to the DNA and causes those growth genes to be. So that's a picture of the DNA right there. You can see the double helix winding around, and that would be a transcription factor. Okay. So I talked a lot about this. I think we can fly through these last few slides a little closer. I mean, a little faster without all the explanation. I hope I've set enough of the background up that I haven't confused. So getting to what we know now about how phytochrome works, okay? So there's one molecule with two sides, like a heads and a tails, two sides of the same coin. So we call the one PR. So phytochrome is receptive to both red light and far red light. And so you see that there's a PR and there's a PFR. When PR is exposed to red light, it changes to PFR. And when PFR is the active form that elicits the response in plants. So when PFR is exposed to far red light, it changes back to the PR. So as long as there is red light, it's all converted to PFR. and it's in the active form. And it's doing what it does. Okay. Now you say, wow, well, that's like 8 to 15 hours in a day, right? Yeah, that's a lot of light. That's a lot of response. And all activated. How is that regulated? Well, in the same way in which we learned several weeks back, protein degradation is the primary way this is. So ubiquitin, again, I want you to notice some common terminology here. The in means it's a what? Come on, just play. Protein. Anything that ends in or A's is a protein. So this is another protein, ubiquitin binding site. And the ubiquitin protein link. So check this out. What we have found now through molecular techniques is that on the PFR version of phytochrome, there's a domain that actually is recognized by ubiquitin. So it's like, I'm a protein and I'm going to express for the destruction of myself. That's what it does. There's just so much of this. It's fine. Trust me. We have life. So on the phytochrome itself, there's a section that is recognized by ubiquitin and through phosphorylation, right? It's tagged for death. It cannot do what it's supposed to do in this form. It gets sent through that proteasome, which you may recall, just chops it up and degrades the whole phytochrome complex. Don't worry, we're going to make some more. All right, so the other thing then is as the sun sets and we go to far red light, once far red light starts hitting this, far red light has, like on the chlorophyll, an effect that it changes the shape and becomes PR. And PR is the inactive form, and so it kind of sits and waits for the next day. Or until as soon as red light hits, it's going to convert back and get going and do it again. So those are the two versions of phytochrome, and we call them, actually there's a lot of versions of them in different plants. I think we've identified 5. So there's PHYA, PHYA, PHYB, PHYC, PHYA, A, B, C, D, E. So PHYA, PHYA, PHYB, PHYC, PHYD, PHYE. Okay. All right. So a little deeper into the rabbit hole. So photoconversion of phytochrome chromophore. Chromophore are those pigments and molecules that are responsive to light and they give color. So a common question then is, well, how does light do that, right? How do those different wavelengths of light cause a different change? And what is the change that's occurring? So this is really neat. And it allows me to teach you a little more basic biochemistry. So this is all good for review. I don't want you to stress out or freak out, because I want you to add it to your memory. So amino acids have an amine group, and they have the carboxyl group, okay, which it's like that. So this is acid, amine, amino acid, and this is whatever it's going to be. And a whole series of these makes up the protein. So I want you to look on here. And so because there's no inherent direction, up, down, back, forth, we have to assign those labels. And so it's always going to be this will go O, and this H will come here, and then there'll be another. We have the peptide bond. You see what's happening? So in this dipeptide here, we're going to have a bond that's going to form between these two. We call this the N-terminus, the terminal, the end is the N-terminus. And we call this side of the amino acid strand, you know, like 100 amino acids later, we call it the C-terminus, the C-terminal. N-terminus, C-terminus. So we have directionality. So here's our N terminus, the NH2. And here's our C terminus, the COOH. You ready? We have the COOH. COOH. In this case, this NH comes from here. And just study that picture for a moment. And this is the whole difference between PHYA PR and PFR. So don't worry about the actual whole big shape of the protein. We're representing the protein now by just a straight line and showing you the amino acid sequence, proline, histine, serine, so forth. Right? So we actually know the amino acid sequence of the phytochrome molecule right here. And attached to the cysteine amino acid is a sulfur, a covalent bond. Right? And then here is the chromophore. This is the light receptive end. So you have this big protein, and this is what chlorophyll is, a protein with a porphyrin ring, right? It's kind of like a protein, but not a protein. Same thing here. Kind of like a protein, well, it is, but it's combined to this chromophore that, check it out, when red light hits it, excites it, and it simply does this. Are you ready? Watch the hand carefully. Same molecule, right? Same molecule. It just, in the trans or cis, we call them isomers. Mer means piece, iso means same. It's the same piece. It's just trans, it's just flipped. And that's all the light does. That's all red light does. So it's crazy to think of these phototropic events in all plants is now reduced to red light striking the phytochrome region and causing it to flip from active to inactive. Now, what makes one active and one inactive is its ability to attract what? Phosphate. So here's a picture of the same thing in a line, a little curve in the rabbit trail. So this is pulled out of Nature Communications. Check out when this was. And this is recent stuff, right? So in addition to regulating a number of transcription factors, phytochromes regulate other aspects of transcription, post-transcriptional regulation, and translation. And a genome-wide study indicates that phytochromes potentially regulate alternative promoter selection of more than 2,000 genes. So here is my mistake sometimes of simplifying. Oh, phytochrome does it. So phytochrome turns on all those genes. Phytochrome is not the protein. It's not even the gene. It's just the receptor and the transducer. But whoa, what it does control is most likely, and I don't think I've introduced this term yet, is master genes. In itself might be a master gene. And what a master gene is a gene that controls other genes. So it's not that phytochrome has to turn on this whole slew of 2,000 genes. It turns on the masters, which in turn turn on. So this is interesting to just knowing the way life functions. Would you really want that many light? When plants depend on light, they thrive by light. That's their livelihood. All to be dependent on one gene? And if it mutates, you're dead? Heck no, right? So you want to be able to have a little more regulation. Let's divide up the system of labor. If I can't do this, at least maybe I can do this, or at least maybe I can do this, or at least maybe I can do this. So of course, once a master gene is mutated, oftentimes you will have major loss of effect in life and so on and so forth. So those things do happen. You know, to be, I mean, not every seed germinates, for instance, you know, and why? Affected, maybe. Virus, nematode, maybe. Environmental, maybe. bad DNA, maybe master gene mutated, sure. You know, who knows why? But every tree, crop, flower has a germination rate. And that's part of breeding and crop science, is trying to get a plant that has a really high germination rate, right? Okay, side note. Anyway, that's pretty cool. So we've heard of alternative splicing for proteins, but I just read to us about alternative promoter selection. So somehow it selects which promoters to bind to and which light genes to turn on, or which master genes to turn on. So here is more to the story. And this is, let's just go slow right here. I want you to realize what we're looking at. Because for, I don't know, 85 years, from 1900 to 1985, one gene, one protein hypothesis. That's what it was. Enzymes are so specific for what they do, like the kinases, right? Your only logical conclusion is there's a single sequence of DNA that codes for that protein to do that function. And what we see right here is a protein that actually has a protein binding site. We already know it has a ubiquitin binding site and a protein kinase. So wait, it's adjuncted to another. I think Hunter brought it up once, but the example I gave back when was hemoglobin. You have 4 of the same polypeptides put together to make one big protein. So that's what phytochrome turns out to be, a really large protein. This right here, the kinase, is just one part of this very large amino acid sequence. Again, here's the N terminus, here's the C terminus, and this is the apple protein, which means not active yet. And then there's a chromophore attached to it. Where was that chromophore? It was on this cytosine or cysteine region. So within that amino acid sequence, there's a specific location where the chromophore is located, and you have all these other domains within the protein. Woo. I mean, you knew it had to be robust, right? Because we just learned that we have 2,000 genes that are ultimately controlled by this. So how does phytochrome work? We got to remember that PFR can enter the nucleus, but PR cannot. So only under red light conditions, PFR can get into the nucleus, binds to transcription factors of a light-regulated gene. So this gene is only turned on when there's light. Here is the red light. How does that gene know when there's light? Right? Phytochrome. So activated phytochrome comes in, binds to the transcription factor, and here's the protein binding site. The protein binding site. And yeah, those transcription factors, which are just wandering around the nuclear cytoplasm, finally get activated only when PFR is present. And now light response, and that could be what? Growth, the phototropic response of auxin. Slightly more detail. So I told you about phosphorylation and kinase, and it usually does this to other things. This is not doing it to other things. This is doing it to itself. And it just gets more. So there's that kinase, and I showed you on that long molecule, there's a protein binding region, chromophore, kinase domain, protein. So anyway, here is that. So we know we have the transcription factor domain, we know we have the ubiquitin domain, and now we know we have a kinase domain, which auto-phosphorylates itself. So this is like, done. And as soon as it's phosphorylated, it's in its active form, and it phosphorylates, it allows it to bind to transcription factor. You can see here that it's released from the block. Now, this is another regulator protein that's blocking transcription unless what? Activated PFR is here. When activated PFR is here, it phosphorylates it, releases the transcription factor. This guy says, I'm not letting go unless you give me, oh, I like that. All right, phosphate group. So he tags him with a phosphate group, he releases this guy who's able now to freely bind. This is a different way. Did you see that? Yeah, there are two different ways now that phytochrome is able to act. One is directly with the transcription factor, and the other is, by activation of other transcription factors for other light response genes. Questions? Is this getting repetitive yet? In one sense, the more repetition of some of these terms, the better, I think. But then I got one for you. So the hint is on the slide, this one here. So we saw two different ways phytochrome acts. So if the protein kinase region, if this region right here is, this region right here is mutated, will that stop all light response? And the answer is no. It would stop a lot. But remember the light response of phytochrome, there's cell elongation, right? Growth, germination, whatever the various functions possibly of auxin are. So these functions right here, yes. If this kinase region, the protein kinase region is mutated, whatever this gene is, you're right. That would probably be stopped and a lot of stuff would not happen, but it would not stop everything. This robust molecule is this way because again, it controls about 2,000 genes and sometimes just being in its active form of PFR is enough to turn on a lot of different genes, okay, for light response. So yes, it would stop some, but not all red light responses would stop. I think it's probably enough. The rest, we just have to kind of step back. There's a few more. This is one I wanted to get to as a photo, photo 1, photo 2. Well, I didn't talk about photo 2. So this goes a little bit to some gravitropism, but also you can see another kinase region, the photo 1, another signal transduction pathway. This is the most recent one discovered. But you can see how when blue light hits this, it's not red light. Now we're talking about blue light. This is phototropin. So this one actually earned the name of phototropism, right? Phototropin, it's a protein that responds. So very similar. You still see signal transduction. It's also autocatalytic in that it's autophosphorylates the same way. And more recently, check that experiment out. This is just extra for fun stuff. Love 1 and Love 2, and this is a flavin mononucleotide. So these are things related to tropism. And we found out that they bind to Love 1 and Love 2. Light oxygen voltage. So when these two right here are struck by blue light, right? Boom. It has a kinase domain like phytochrome. It has a special J alpha helix. Don't worry about that. It's just a special helix. It'll be important on the next slide. But, so this is the structure of the protein, phototropin 1. And you can see that we have very nice, strong bend towards light whenever this gene is activated. And so what they did to find out if both of these, kind of like Austin's, I think it was Austin's experiment last time, so we knocked out LOVE1 and we still had light bending. All right, knocked out LOVE2, put it in light and nothing happened. They lost their ability. So phototropin 1 has 2 antenna receiving areas, and it turns out LOVE1 is the more important one. If you mutate, LOVE2 is the more important one. If you mutate it, you don't get any phototropin. You don't get any tropism. So dephosphorylation. So here, notice this is in the dark. Blue light causes, just imagine everything we've already talked about with phytochrome, just a different protein, right? This one's in the cell membrane. And it has a kinase domain, and it's caged. It's locked up. It cannot catalyze itself. You see that? It's bent right here. It's bound up. On the previous slide, it was in a straight line, right? But now it's bound up, it's caged, so long as it's in the dark, dephosphorylated, nothing's happening. But when blue light strikes these, phosphorylation occurs. This is unlocked kinase domain. Whatever that substrate is in the signal transduction pathway can now phosphorylate, and we get the phototropic response. And again, that's going to be receptors for auxin, which signal cytoskeletal movement, cell wall degradation, expansion, all those things, because really growth and bending is a lot. All right, so that's a signal transduction pathway. If we're supposed to put all this together, we'll notice that this is right here, disk space, light striking these. You have all these proteins, which are all second messenger systems. It's a pretty complex picture to paint. Whew. Sorry to put you through that, but you asked for it.